So welcome to Biotechnology for Drug Discovery Session 3. Here in the session we're going to look at producing recombinant products using eukaryotic systems. So the learning objectives for this next session are 1. To be able to describe different eukaryotic expression systems for recombinant protein production. 2. Understand the differences between yeast, insect and mammalian systems. 3. To explore the advantages and disadvantages of each eukaryotic system. So now we're going to look at the different kind of protein expression systems. We've already visited the prokaryotic expression system, but here we're going to actually focus now on the yeast, the insect and the mammalian expression system. So firstly we're going to look now at the yeast expression system. So here's a list of some of the pros of the yeast expression system. They are easier and less expensive than high eukaryotic cells, so namely insect cells using baculovirus and also mammalian cells. They have rapid growth on inexpensive media, so the media is fairly cheap to buy, and you can grow up quite large numbers of yeast. And so they're actually ideal for large-scale production of heterologous proteins. They're often compatible with genetic manipulation, so there's many genetic tools available, and also they exhibit near native conformation concerning like the protein structure of proteins, and also they also possess some post-translational modification, and as well as processing. So, for example, if you get N-glycosylation, they're able to process the N-glycan chain. Or if there's a signal sequence that needs cleaving, they have signal peptidase. They have well-defined secretory pathways for extracellular export, much like other eukaryotic systems, which is really useful if we want to produce protein which is actually secreted from the cell. And usually they're quite safe to use. Now, there's a couple of kind of disadvantages of the yeast expression system. For example, they lack post-translational modification required for specific activities, and there are specific post-translational modifications which are unique to each type of yeast strain. They're a little bit more expensive than the prokaryotic system, but not as expensive as mammalian systems. And often, sometimes, they lack mechanisms for proper folding for some eukaryotic proteins, so they're not always the ideal system to express your protein in. If we compare the growth of the different systems, so for example, if we compare E. coli, we know that it doubles every 20 minutes, whilst yeast double every 90 minutes, so they're a little bit slower, but they're much quicker than, say, mammalian cells. So this is actually a lung cancer cell line that doubles every 18 hours. So at Thermo Fisher, there's a website which you can use to help you design or select the vector. And here we can actually select different host systems, okay? And particularly if we select the yeast host system, that gives us around about 28 products, and these are different vectors which then can be selected according to their type. Also the selectable markers which we use for antibiotic selection, any kind of N-terminal, C-terminal tags, promoters, etc. So this is an example of a yeast expression vector from Piscia pastoris. Many yeast vectors are kind of based on this kind of 2 micron circle of yeast. And the native 2 micron circle has been actually modified in a variety of ways for use as a kind of cloning vector. So they look quite similar to our kind of bacterial vectors, and actually they have components that are kind of which can be used to be propagated in the bacteria. So there's a number of features, things like this promoter fragment, so the IOX1 promoter fragment. When you see this primer site, this is really useful because actually we can use this to to kind of PCR up products. Maybe it's useful for actually detecting by sequencing. There's this alpha factor secretion signal. Again, also you have a primer site as well, which we can use for sequencing. Also, there's multiple cloning sites, so it's here. So we've got SNAB1, ECHO1, and AVR2, and also NOT1. So also we have the 3' AOX1 primer site, and also the transcription termination. There's also a HIS4, and also we have a chamaemycin resistance gene, as well as the ampicillin resistance gene for selection in the bacteria. So transforming fungi. So to transform fungi or yeast, we use something called spheroplasts, which are kind of wall-less cells, which were kind of first developed in Saccharomyces cerevisiae. And what happens here is that the cell wall is removed enzymatically. So using lytic enzymes kind of results in then these spheroplasts, which are then fused with using ethylene glycol, or known as PEG, in the presence of DNA, okay, and calcium chloride. Okay, so this then allows for to generate the new cell wall, okay, in a kind of a stabilizing medium, which then contains 3% agar. There's then electroporation. So essentially we kind of use electric pulses, which allows us then, which kind of is a simpler and more convenient way of transformation of yeast cells. And DNA can actually be introduced into yeast and filamentous fungi also by something called conjugation. So there are different types of yeast plasmid vectors, and this is just kind of a kind of simplified list of those, and we'll discuss about each type. So we've got yeast integrating plasmids. Yeast integrating plasmids are kind of the most, are in most cases suicide vectors. So this means that the vectors are unable to replicate in the destination host, and they actually become integrated. Okay, so they get integrated and then disappear. So essentially any plasmid that can be efficiently transferred into the recipient. So what they do is they lack an RE site, and so they have to be integrated directly into the host chromosome. And so they're integrated between this MAT alpha 1 and MAT alpha 2 via a homologous recombination, which is for a kind of efficient multiplication. We also have the yeast episomal plasmids. They were actually first constructed by Beggs in 1978. So this was by recombining an E. coli cloning vector with a naturally occurring yeast 2 micron plasmid. And so the word episomal indicates that the YEP can replicate as an independent plasmid, but also it implies that there is integration into one of the yeast chromosomes that can occur. We've also got yeast replicating plasmids, so YRPS. These vectors contain an autonomously replicating sequence of ARS, which are kind of derived from a yeast chromosome. So as the name kind of suggests, they can actually replicate independently of the yeast chromosome. However, they tend to be quite unstable and could be lost during budding of the yeast. We've also got yeast artificial chromosomes. We've actually come across these before in molecular biology. They are artificial chromosomes. There are two forms. So you have the circular form, which is for growing in the bacteria, and also you have a linear form for growing in the yeast. The circular form is maintained like any other plasmid in bacteria, but the linear form must have kind of telomere sequences, and this allows it to be maintained in the yeast. The linear form can actually hold up to over 2,000 kilobases of cloned DNA, and this is particularly useful for genomic research. So there are different types of yeast. We've got three main types. You've heard of the Saccharomyces cerevisiae. I've also talked about the Piscia pastoris, and we also have the Schistosaccharomyces pombe. The Saccharomyces cerevisiae, they're also known as baker's yeast. This is what they look like under EM. They are a single-cell eukaryote, and they grow fairly rapidly, as we've seen on the graph. They have a doubling time of approximately 90 minutes. They prefer to grow in simple defined media, so such as YEPD, and many, but not all of the post-translational modifications. They have strong constitutive promoters, promoters like phosphoglycerate kinase PGK or glyceraldehyde 3-phosphate dehydrogenase GPD, and also the alcohol dehydrogenase ADH1. They also suffer similar problems as constitutive E. coli expression systems. They have inducible production, so they via something called a GAL1 system and a CUP1 system, which we'll talk about now. The GAL1 system is a galactose is actually converted to glucose 6-phosphate by enzymes called the Lalore pathway. The lower pathway is kind of structural genes known as the GAL genes. They're expressed at a high level, normally between 0.5 to 1% of total cellular messenger RNA, and galactose as a sole carbon source. The GAL genes promote a site for a transcriptional activator called GAL4P. So the galactose-inducible gene expression in yeast, the expression of genes from kind of multi-copy vectors under the control of GAL1 promoter can be increased substantially if the genes are encoding a transcriptional activator known as GAL1 and GAL4. So GAL4 is also placed under the control of GAL1. In this case, induction by galactose will actually produce more GAL4, and consequently there'll be more production of target protein. So this is a good way to kind of increase protein expression in the yeast using the galactose-inducible gene expression system. Another type of gene expression system is known as the CUP1, and the CUP1 essentially is it kind of uses copper metabolism in the yeast, and copper ions, so known as copper 2 plus or copper plus, are essential but can be toxic at high levels. Saccharomyces cerevisiae, they have copper homeostasis, so uptake through these special kind of channels and distribution and detoxification mechanisms. So at high concentrations, there's a detoxification. It's mediated by a copper ion sensing metalloregulatory transcription factor called ACE1P. ACE1P recognizes when there's a kind of high level of copper. And actually the copper is kind of targeted to the nucleus. When you have an interaction of copper with ACE2, it binds to kind of DNA upstream of the CUP1 gene, which encodes this metallophilin protein and induces transcription. So the expression vectors which harbour the CUP1 promoter can induce target gene expression in this kind of copper-dependent fashion. And so you can grow, this can then be grown on rich carbon sources such as glucose to high cell density, and then protein production can be initiated by the addition of copper sulfate, normally at about 0.5 millimolar final concentration. So this helps to kind of increase the levels of protein production. There is obviously a drawback, so the presence of copper ions can be found in yeast growth media and also in certain water supplies. So if you have water which is distilled in the lab, it may also contain copper, and so this may not be ideal. So now moving on to the next yeast system or next yeast strain, known as Pichia pastoris. Pichia pastoris is a methyltrophic yeast, and this is what it looks like under the electron microscope. It looks a bit like I don't know, kiwis maybe, or limes. Quite pretty. It's obviously been coloured. It metabolizes methanol as a carbon source, and the metabolism of methanol, you get oxidation of methanol to formaldehyde using the molecular oxygen by the enzyme alcohol oxidase. The alcohol oxidase has a poor affinity for oxygen, which compensates for generating large amounts of enzyme. And the promoter, which is kind of regulating the production of alcohol oxidase, OX1, can be used to drive the heterologous protein expression. And we saw in the vector before it has the OX1 promoter. So P. pastoris cells, containing the expression vector, are integrated into the genome as a single or multiple copy. They can grow in glycerol, so glucose, which represses the AOX1 transcription, even in the presence of methanol, to a kind of extremely high cell density. Now you can induce target protein expression by the addition of methanol, and you can get anything between 0.5 to 10 grams of protein per litre of yeast culture, so that's quite a lot. The expression of gene encoding recombinant, say hepatitis B surface antigen production of more than 1 gram of antigen from 1 litre of P. pastoris cells. The P. pastoris may have an advantage in the glycosylation of secreted proteins. And glycoproteins which are generated in the P. pastoris are more closely resemble the glycoprotein structure of those found in higher eukaryotics. So this kind of just summarizes the expression systems in the P. pastoris, and that the secretory pathway, they have secretion signals. They also have prokaryotic processing, as well as glycosylation, both N and O glycosylation, and as well as membrane associated. They can get membrane associated proteins or surface display anchors. Okay, so Saccharomyces pombe. The properties are similar to those found in high eukaryotic organisms. They have chromosome structure and function, and also cell cycle control, as well as RNA splicing and codon usage, and they're an ideal candidate for production of eukaryotic proteins. They can have folding which is proper, folding properly, which may reduce protein insolubility. Okay, so protein production can be controlled by the expression of something called NMT1. This is where there's no message in thymine promoter. They're active actually when the cells are grown in the absence of thymine. So here we can see an example. So this is the bright field, so showing the cells on the plate, and then this is the GFP fluorescence. So in the presence of thymine, the expression of the GFP is fairly low, it's very dull, whilst in the absence of thymine, what we see is there's an induction, so allowing downstream transcription of genes which are under control. So in the presence of which then kind of in the absence of in the presence of thymine, there's a very low GFP signal, and in the presence but in the absence there's a very high level of GFP fluorescence. So there are a number of selection markers that can be used in yeast, and these are particularly useful for different varieties. So, for example, the URA3, it's a pyridine or uracil. It has counter selection, yes, when grown with something called 5-FOA. This is used in Saccharomyces cerevisiae. Yes, this can be complement for something called URA4S-POMB, but the complementation is weak. There's like different ones you can see. We've mentioned tryptophan before in bacteria, but they also appear in the Saccharomyces cerevisiae. I won't spend too much time on these. Okay, so moving on now to insect or baculovirus expression systems. Baculovirus is kind of classified as group 1, so it's a double-stranded DNA. So the double-stranded DNA is transcribed into messenger RNA. It's a family of the Baculoviridae. Its subfamily is known as the nuclear polyhedros virus, or NPV. And there are different genera. There's alpha baculovirus, beta, delta, and gamma baculovirus. The baculovirus structure, the virion actually exists of two forms. It exists as a budded virus, so this one here, which it's where the neocapsids are budded from the host cell's membrane and are involved in a kind of secondary infection. It also exists as a kind of polyhedral hydra-derived virus known as PDV, which are kind of nuclear capsids which are packaged into a kind of polyhedra or an occlusion body. These are kind of stable in external environments. So there's kind of two viral forms that exist. The genome of the baculovirus, it's circular and also double-stranded DNA genome. There are a number of small repeated sequences known as the homologous regions, so HRS, which are kind of interspersed within this genome. The HRS they help to enhance early transcription and also act as origins of replication. There's a virus called the Autographia californica, which is a multiple nuclear polyhedrosis virus called MMPV. So the host range of the baculovirus, it's kind of species-specific tropisms among the invertebrates with over 600 host species. So in the immature, so you've got the larval forms of the moth species. They are the kind of most common hosts. But these viruses also have been shown to be infecting sawflies, mosquitoes, and shrimp. But they don't replicate in mammalian or other vertebrate animal cells. The replication cycle, so the life of the baculovirus. So first you have the viral infection. Okay, so this involves the ingestion, the uncoating of the polyhedral coat, the fusion with the midgut cells. Then you have viral replication in the nucleus, and then there's a secondary infection. This is where the budded virus is released to infect systemically. So there are a number of kind of viral genes which are important in the baculovirus. So we have, first ones are the immediate early, so this is the expression of viral transregulators and genes which do not require transregulators. We also have the delayed early, so these are its expression of genes involved in the replication of the virus and manipulation of the host. We also have the late, which are characterized by the shutdown of the host cell DNA replication and protein synthesis, and the baculovirus is produced, and this disseminates virions that become occluded in protein polyhedron. And then we also have the very late. This is the viral proteases, which help to liquefy the host and degrade the chitinous exoskeleton, occluded progeny virus, and then released onto the surrounding material for horizontal spread. This guide, which was available from Invitrogen Life Technologies, now kind of obviously owned by Thermo Fisher, but you can find this at the Thermo Fisher website. It's an excellent guide to baculovirus expression vector systems and also insect cell culture techniques. It's worth downloading and having a look at because there's some really nice information in this guide. So we're going to now explore different parts of the baculovirus system and how we can use the vectors to kind of in protein expression. So baculovirus as an expression vector. So there are many non-essential genes which can be now replaced by a gene of interest. The resulting baculovirus lacks one of the non-essential genes, such as POL-H, VCAF, GA, which can be replaced with a foreign gene. And also there are powerful viral promoters known as the late, very late, and the late and very late phase genes. The construction of the recombinant ACM-MPV involves the transfer vector method. So you co-transfect expressed vector with the second plasmid, which contains a kind of required viral genes. So these are co-transfected to produce the virus, which is then kind of amplified. So that's one system, so that's the transfer vector system. And then there's also one which is called the back-to-back system, which you transform E. coli, which contain the BACMID DNA, and then they directly infect insect cells, which produce a virus and then amplify the virus. And there are three cell types which are generally used, known as the SF9, SF21, and Hi5. And these have late very promoter, known as the P10, plus the polyhedron, which drives the expression in the late stages of the virus cycle. So for homologous combination, we can use this transfer vector known as the PVL1393 or the PVL1392, which has a size of about 9.8 kilobases. So we can use a transfer vector as well as our cloning vector, which we can then recombine as the recombinant transfer vector. This then can be transfected into it can be then combined with the baculovirus. So there are a number of key features. So you've got multiple cloning sites. There's also recombination sequences for insertion into the baculovirus genome. There's also the polyhedron enhancer promoter, and then for selection, so for the antibiotic selection, we can use the ampicillin resistant gene in the bacteria. So for generating recombinant baculovirus by homologous recombination, there are kind of like a number of steps. Essentially, you start off where you need to clone the gene to be expressed into the transfer vector, and on day 0, you need to then co-transfect insect cells with the wild-type ACN-MPV DNA and the recombinant transfer vector. So it's two vectors that we need to transfect at the same time. At day 5, we then, from a low titer, anything between 1 times 10 to the 2 or 1 times up to 1 times 10 to the 4, we kind of generate a recombinant viral stock, which then we kind of collect the virus, so it's secreted into the media. We purify the desired recombinant with 3 rounds of it's called plaque assays, which we'll come to in a moment. Afterwards, by day 26, we can then amplify the virus. So there's like 2 to 3 rounds to amplify and build up a big stock of virus, of which by days 40 to 47 we get like a high titer, so anything between 1 times 10 to the 7 or 1 times 10 to the 8 viral particles per milliliter, which is kind of the recombinant baculovirus stock, which then can infect the cells. And then by days 43 to 50, then we can express our protein. This actually shows you like this is in kind of a time course for the expression of like fluorescent proteins. In particular, we've got GFB and DS-RET using the SF9 cells, which have been infected with recombinant ACM and PVs, which are carrying the fluorescent markers. These images were kind of taken at 8 hours, 24 hours, 48 hours, and 72 hours post-infection using the fluorescent microscope. And what you can see is there's an increase in expression by 48 and 72 hours post-infection. For the generation of recombinant baculoviruses and gene expression with back-to-back expression systems, it's a kind of faster approach now for generating recombinant baculoviruses. It uses specific transposition known as the kind of TN7, and that helps to insert foreign genes into the BACMID DNA, which is then propagated in the E. coli. The gene of interest can be cloned into something called a fastback vector, and the recombinant plasmids are then transformed into these like DH10 back E. coli cells, which then contain both the BACMID, okay, and also the which has this kind of mini ATTN7 target site with a helper plasmid. So a number of steps. I'm going to go through these steps. I just kind of briefly was talking about them before. Steps in the recombinant baculovirus production. So first we need to clone the gene of interest into our PFAS-back donor plasmid. Then the expression cassette in the PFAS-back is then flanked by, at the left and the right arms, with this TN7. The cloned PFAS-back is then transformed into the E. coli, which then contains the baculovirus shuttle vector, BACMID, which having the ATTN7 target site. There's a transportation occurs between the mini ATTN7 target site to generate a recombinant BACMID, and you can have PCR application using the M30 forward and reverse primers. So site-specific transposition with the TN7. So here we have the TN7R and TN7L and the gene of interest, which is inserted into this PFAS-back. We transform the recombinant plasmid into the DH10 back E. coli cells, which contain the BACMID. Okay, the mini TN7 element on the PFAS back plasmid can transpose to the mini excuse me, ATTN7 target site on the BACMID. The helper plasmid kind of facilitates the transposition by a kind of transposition protein. So you have this helper, you've got the BACMID and the donor. The transformation of the recombinant plasmid, you get the identification through antibiotic selection, and there's also blue-white screening using the beta-gal. So the beta-galactose, so that's the protein that can be encoded by the LAC-Z gene of the LAC operon that exists as a kind of homotetramer in its kind of active site. And we can use this to select for kind of positive and negative colonies. And so we can see, like here we've got the blue and the white colonies. So we can culture our bacteria, culture our bacteria on these X-gal with IPTG. Along with, we've also got different selectable markers, so chemomycin, gentamicin, tetracycline. The white colonies is where X-gal is not hydrolyzed, so the presence of an insert in the lac Z. So if we have white colonies, that means we have a positive colony. I forgot to mention that this is more specific for the system, so the isolation of the back mid DNA from the DH10 back can be done with this concert high purity plasmid mini prep system, and it's very specific for isolation of this DNA. Okay, so the next part is kind of the transfection. So the transfection of the SF9 cells, and the SF9 cells are grown in 6-well plate, normally at a density of 9 times 10 to the 5 cells. They're normally grown with the media, so here we would grow them with SF900 to SFM, so 2 mils in each of these wells. They're incubated for 1 hour at 28 degrees. And then we prepare a kind of transfection mixture, so which contains our back mid DNA, so 5 microliters in 1 tube with where we add then SF900 to SFM media, which is up to 100 microliters in one tube, and then in the second tube, tube B, we add the self-vectin, which is a bit like a transfection reagent, around about 1.5 to 9 microliters, depending on how much DNA we use in the first tube, which then we add also, we dilute in the media, and what we do then is we mix B and A together. They're kept at room temperature for 15 minutes, and this helps them. It helps the formation of a lipid DNA complex, essentially a bit like a liposome forming. So essentially, the DNA is taken up inside micelles, which are formed during this process. Afterwards, we then dilute this transfection mixture using the media, so the SF900 media in 0.8 mils. So that would then make it up to then 1 mil. And then we then overlay the, so kind of slowly add the mixture to each of these wells. And the cells would have been washed, and then you would have added fresh media. Then you incubate then for 5 hours, so it's quite short actually compared to mammalian transfection. So normally you would incubate overnight for at least 18 hours. So you incubate for 5 hours at 28 degrees, and then after which time you remove the transfection mixture, then you add fresh media, and then you incubate then for 28 degrees for 72 hours. After which time then you can then harvest the virus at 72 hours post-transfection. So we're going to discuss now the virus plaque assay. So the infectious potency of a stock of baculovirus can be determined using an immobilized monolayer culture using kind of agarose overlay. We can identify plaques, so wild type, decreased cell density, we get enlarged nuclei. Many have large, dark, angular occlusions bodies in the nuclei. So this is something we can observe like under the microscope. We can also perform, if we're looking at recombinants, we get like milky grey plaques. So you can see here there's some milky grey plaques, and these have been kind of like diluted. You have small or low contrast, and you can get staining with neutral red solution. And so by recombinant expressing it's like chromogenic markers, so we can use things like luciferase, beta-galactosidase, or blue gal or X-gal. So the insect cell culture, these are invertebrate cells. The insect cells grow at anything between 25 and 28 degrees C. They prefer a pH growth meter of between 6 to 6.4, so that's really important to understand, and also we may need to monitor that if we're doing a bioreactor. They are non-carbon dioxide, have a non-carbon dioxide equilibrium, and they're a kind of open-capped culture system. It has an osmolarity within 345 and 380. So there are different cell lines that are available. So these are some of the common ones, so known as the SF9, SF21, from the Sporodopteran Fruga perda. We also have the TN368 and the Hi5 from Trichoplusia near. This is an example of the SF9, of what they look like under the microscope at different densities. You remember this from last year when we were doing the CDI project and we were looking at cell lines. Now this information is available from ATCC. So at kind of low density, you can see what they look like. They either grow in suspension or they adhere. And this is what they look like at a kind of high density. So they kind of clump up. They like to form these clumps as opposed to growing kind of in kind of single colonies. So the recommended conditions, so things like cell density, anything of around about 2 times 10 to the 6 viable cells per mil, with about 80% confidence. The culture vessels that you can use Erlenmeyer flasks, which contain different volumes. So if you're using an Erlenmeyer flask of 125 mils, then you use a volume of 35 to 50 maximum. And for 250, anything between 75 and 100 cell suspension respectively. If you're using flasks, so if you want kind of have 80% confidence, you use a T75 flask or a T162 flask. Total volume is anything between 50 and 20 for the smaller flask, 40 to 50 for the bigger flask. The C density, so we want to use like anything between 3 to 5 times 10 to the 6 cells per mils. And for this one, we want to use anything between 2 to 5 times 10 to the 4 cells per centimetre squared. The incubation conditions, so we want to use a temperature of about 28 degrees, non-humidified, meaning that we don't add water to the incubator. It has an ambient air regulated incubator or warm room on an orbital shaker, anything between 125 and 150 RPM. And then in the flask culture, we have anything use a 25 degrees non-humidified ambient air regulated incubator. So we've mentioned that we have different types of insect cells, and they all have different kind of doubling times. So the SF9, they are probably the slowest, slow growing ones. And the SF21 has 24 hours doubling time, and the Hi5 have an 18-hour doubling time. The shape, as we saw in the pictures, they're spherical, granular, regular in size. The SF9 like to attach to a surface along with the SF21, whilst the Hi5, they actually will loosely attach to the surface. They grow in different media, so that's something to bear in mind. This is obviously a media which is made specifically for these cells. The origins we've already discussed, particularly the Hi5, they are ovarian cells of cabbage looper. The types of cultures, they mostly grow as a monolayer, but also they can grow as suspension for most. So the advantages of back-to-back expression system over the homologous recombination. So it's a one-step purification and amplification. Higher purity of isolated recombinant viral DNA, so it's not mixed with parental or non-recombinant viruses. And you can also get a rapid simultaneous isolation of multiple recombinant viruses, and the advantages of back-to-back expression system over homologous recombination. Okay, so mammalian cell expression systems. These are important for producing proteins with all post-translational modifications. And many established cell lines are useful. We have transient expression, so things like African green monkey, baby hamster, human embryonic, all kidney tissue cell lines. And long-term expression, we use Chinese hamster ovary and mouse and melanoma cells. The expression vectors in these systems usually are derived from the animal virus such as SV40, simian virus 40. They can be used for expression of single polypeptides, homologous, heterologous. The latter is made possible by transforming with two or more separate clone genes, so like we saw in the insulin story at the beginning. Industrial production is, however, costly, and that is a kind of downfall of mammalian cell expression systems. There are a number of established mammalian cells. For example, we got Chinese hamster ovary. There's an epithelial cell line derived from the ovaries of Chinese hamsters. We've also got DG44. It's a Chow cell line, but has a marker sign called DHFR. We've got duck B11. These are also Chow cells, but have a marker of GS. We've got NSO, which is a myeloma cell line derived from beta lymphocytes of mice. We've also got HEC293. These are a kidney cell line, an epithelial cell line derived from the human embryonic kidney cells, which have been transformed with the adenovirus DNA. Other cell lines we have available are the BHK. This is a cell line derived from kidney cells of baby Syrian golden hamsters. COS cells, they are fibroblast cell line derived from the kidney cells using SV40 transformed of the African green monkeys. And we've also got PERC6. It's a trademark cell line derived from the human retinal cell developed and known by a company called Cruzel Holland BV. The transient gene expression, so cell lines which are used for transient gene expression are things like the COS cells. They're particularly great for this. They tend to produce a lot of protein, fantastic for transient, and you get really high expression level in the COS cells. The human embryonic kidney cells, these are HEC293s. They're also really good for kind of transient gene expression. Transient gene expression is actually a convenient method for rapid production of small quantities of protein, and this is for initial kind of characterization. So if you want to just check to see how your protein is being expressed before you go ahead and make stable cell lines, because making stable cell lines can take a long time and effort and also a lot of resources, and it can take up to 3 or 4 months to establish a stable cell line. It's a rapid way of testing the vector functionality, as well as the optimization of different combinations of promoters, of elements in expression vectors, so you can see which ones are going to work best with your particular gene of interest. Looking at stable gene expression, there are a number of cell types which are used for stable gene expression, in particular the Chow DG44, the Chow K1, the PERC6, NSO, SP2, \0, and BHK. Now, they're particularly good for stable integration of plasmid into the host genome, or the host chromosome. They're used for long-term stable gene expression, and this is when high yields of heterologous proteins are required. And there's been around about 140 recombinant proteins that are currently approved for therapeutic use, which mostly use the Chow cells to produce them. If we look at the vector design, so the first vector was based on this simian virus that can replicate in several mammalian species. Its use is restricted to small inserts, and that's because they're only limited to the amount of DNA that can be packaged into the viral capsid. Other vectors that can accommodate larger amounts of cloned DNA So we've got the adenovirus, which can be maintained as a multi-copy plasmid in some mammalian cells. And also we've got the adeno-associated virus, which can integrate into specific sites in the host genome. Kind of more generalized expression vector. So this is quite a famous one. It's called the pcDNA5 TO or TOPO. There are a number of kind of common features in this vector. In particular, it has a promoter, so the cyclomegavirus promoter. As I've told you before, it's a very strong promoter. This vector actually is used for inducible expression, so it's also got this tet-on, tet-off system. So you can use tetracycline to induce the expression of your gene. There is many restriction enzymes available for cloning, so this is a multiple cloning site. You've also got the hygromycin for selection in the as a stable expression, and also you have ampicillin for replication in E. coli. The features of these vectors, so for best results of a gene interest, you must be equipped with translation control sequences. This kind of, the gene of interest can be fitted with various sequences that enhance translation and facilitate both the secretion and also the purification. We have things like the COSAC sequence. This is kind of a specific sequence surrounding the AUG, so it's the start codon, a signal sequence, and also the protein affinity tag for purification, also proteolytic cleavage site, and stop codon. And also the 5' 3' UTR increases the efficiency of translation and contribute to messenger RNA stability. Here, this is kind of a schematic showing different mammalian expression vector components. In A, this is an expression vector which contains a promoter with or without something called an enhancer element. You also have a 3' polyadenylation sequence and also something called a GOI and monoclonal cloning site. There's also a mammalian selectable marker, which is kind of separated by the it's a separated set of kind of regulatory elements. The vector B, it's known as a bistromic vector, which contains kind of a single cassette of expression of a GOI and also multiple cloning site. And the a mammalian selectable marker, okay, it's kind of separated by this IRES, which is under control of the upstream promoter, and also you have the 3' poly A. In vector C, so vector C is an expression vector which contains kind of DNA opening elements, and then we also have vector D. So vector D is an expression vector that contains one or more like introns, one or more intron exons, which can be inserted into a kind of coding sequence for a gene of interest. Focusing more on the expression systems, so there are different types of expression systems. So we have the 2-vector expression system. For example, we saw with insulin which was expressed in bacteria, but also this also exists in mammalian systems. So where we can separate different genes onto different vectors, and then we can purify and then recombine a kind of expressed complex. We've also got the bistromic, so bisystromic IRS vectors. These are kind of containing, allow kind of simultaneous expression of 2 proteins separately, but from the same RNA transcript. So how does this work? Well, the IRS is it's known as the encephalomyelitis virus, so ECMV. This allows translation of 2 open reading frames from one messenger RNA. So the translation allows the expression of the gene and also a fluorescent gene. In the case we could have GFP or DS-RED, for example. And so what happens is then you generate actually 2 products. So you get your protein of interest, but also you can get expression of your fluorescent protein, which is expressed separately, but is also found on the same vector. So there are a number of selectable markers for mammalian expression, which we can use. For example, we've used these are metabolic selectable markers, so known as dihydrofolate reductase. We can use the methionine selectable marker of sulfur hexamine, known as MSX. We can also use the glutamine synthase, GS, with the selectable reagent of methotrexate, MTX. There are a number of antibiotic selectable markers, including puromycin, blastocidin, histamine, hygromycin, ciamycin, bleomycin, and neomycin. Focusing now on DHFX, MTX gene amplification. Resistance to methotrexate can occur via 3 different methods. So we have methotrexate permeation mutation, including MDR. We can have altered DHFR with lower methotrexate binding affinity, and also we can have overproduction of DHFR protein. The human DHF, this is essentially forms a conformer, which kind of preferentially binding the DHFR messenger RNA. The human DHFR conformer actually preferably binds to NADPH, and in the presence of methotrexate it frees up the messenger RNA, and this actually increases DHFR protein. So engineering mammalian cell hosts for enhanced productivity. We can actually improve self-growth and viability under different culture conditions in the bioreactor, and this is to kind of prevent P53. So remember, we've looked at P53 and its kind of role in the cell cycle, and the P53 is able to kind of activate cell death and cell survival from kind of activating the response pathway. The mouse double mutant known as 2 protein, which is known as the MDM2, can bind to P53 and prevents it from acting as a transcription factor, so it gets degraded quite quickly. The MD2 also marks P53 then for degradation. So what happens in the absence of MDM2 is that P53 will act as the transcription factor and will induce activation of various genes, but we can use this as a way to kind of stop cell death in our kind of cell cultures. So we can engineer the mammalian cell hosts for enhanced productivity. So P53 and MDM2 can form this kind of feedback loop, and under kind of cellular stress, so such as DNA damage, P53 is actually stabilized, and so the increasing P53 activity and resulting upregulation of P53 target genes, and that helps to obviously promote tumor suppression activity. The P53 mediated activation of MDM2 transcription generates MDM2 protein that kind of causes this MDM mediated ubiquination and then subsequent degradation of P53. We also get low oxygen levels, and potentially we can form lactate. And so under low oxygen conditions, many cells, including the Chow cells, they will secrete acidic waste product, lactate, as a kind of struggle to obtain energy from the glucose. So the glucose, which is like converted to the pyruvate, under anaerobic conditions, the pathway will be pushed towards the lactate by converting pyruvate to lactate with lactate dehydrogenase. Under these conditions, the pyruvate is actually the intermediate compound that's produced during the metabolism of glucose, and it's converted to this lactate. And it does this in preference to entering the TCA cycle. And the pyruvate carboxylase has a lower level of activity in the absence of oxygen. So, you would lead to kind of a buildup of lactate in the media. So there are a number of advantages of the mammalian system. So one, including the post-translational modifications. It has high functionality, and that's because the proteins can fold properly, and also they have sufficient post-translational modification. They're compatible to humans. They have low immunogenicity to humans. The mammalian cells are used other than human cells, are not susceptible to human pathogens, and are highly safe to use. There are a few disadvantages of the mammalian system, including the mammalian cells are actually fragile, so very sensitive to changes in temperature, and also they are also slow growing. So we can see that they are much slower than other systems that we've looked at. The selection takes time, so if we want to make stably transfected cells, it takes 3 or 4 months to establish this. They can be contaminated with animal viruses quite easily, so we need to work in an aseptic situation, but they are also, they are really susceptible. The mammalian cell culture technique is very expensive, so compared to obviously the baculovirus and also to the bacterial systems. And transfection of gene of interest is sometimes difficult to achieve. This is kind of like a final slide, which is just to kind of summarize the different systems and compare things like the cost, the timeline, the yield, the storage advantages, and also the disadvantages and challenges of each system. In particular, the bacteria system, it tends to be the cheaper system per gene, so around about $5,000 per gene, compared to mammalian, $20,000 to $25,000. So you can see how much cheaper the bacteria system is. The timelines that it takes to achieve the expression or to obtain enough protein can be anything between 3 months for bacteria up to 6 months for mammalian. The yield tends to be high in bacteria, yeast, and baculovirus, whilst it tends to be low in mammalian systems. The storage, so we don't really need specialized storage for bacteria and yeast, just in the glycerol stocks, whilst the baculovirus has to be maintained as a viral stock. The mammalian stocks, they need to be frozen. If they're frozen at -80, they can keep for about 6 months to a year. Whilst if they're kept at at liquid nitrogen, then they're being kept for several years. The advantages are for the bacteria system. They are simple, and use inexpensive media. You have high cell density. They are genetically well characterized. The fermentation process is worked out for scale up, and they're FDA approved for standard operation protocols. For the yeast system, they're simple and also inexpensive media. Have high cell density, adaptable fermentation processes. They're genetically well characterized. They do not produce endotoxins like bacteria potentially could. They have high level protein secretion, and they're FDA approved. The baculovirus, the advantages are they have post-translational modification similar to their higher eukaryotes. They have high level protein production. Up to 15 kb inserts can be expressed. They're non-infectious and safe, and they're also FDA approved. Whilst the mammalian systems, they have post-translational modification similar to the native protein. They're safe with no immunogenicity concerns and are also FDA approved. Now, the disadvantages and challenges. For the bacteria system, you have no post-translational modification, whilst with the yeast system there is N and O glycosylation, but the pattern is slightly different to the higher eukaryotes. The baculovirus, the insect cells grow slowly, whilst the mammalian cells, they also grow very slow as well. Kind of just picking out some of these now. The bacteria system, they produce endotoxins, and the proteins can be degraded by proteases. You can also get proteolytic degradation in the yeast system. In the baculovirus, you may get improper folding of a protein at the late stages, and you also may get complex glycosylation, which is not possible. For the mammalian systems, it's actually time-consuming and laborious, and the protein yields, you have very low protein yields, and it's also very expensive. Anyway, so this final slide now is just to kind of indicate some further reading. This paper here is excellent. It's like talking about baculovirus as a mammalian cell expression vector for gene therapy, so that's a really nice resource. Have a look at the guide to baculovirus expression vector systems and insect cell cultures. That's available from Thermo Fisher from this website. Another paper looking at the expression vector cassette engineering for recombinant therapeutic production in mammalian cell systems. This is a really nice paper. It's just been published in 2020. And finally, take a look at chapter 3. This is focusing on mammalian cells in biotech production from Kaiser in pharmaceutical biotechnology, drug discovery and clinical practice. Anyway, that finishes our session today. Hopefully see you in the session.